Today, I will start the new chapter of molecular life science. My name is Takanori Oyoshi. I will focus on the nucleic acid conformation. This lecture is four chapters. First is about the duplex DNA, including A form, B form, Z form structure. Second chapter is triple helix, quadruplex, I motif structure of DNA. Third chapter including local nucleic acid conformation binding protein. For example, duplex and triple helix, quadruplex I motif binding protein. And finally, I will explain about the relationship between local nucleic acid conformation and disease. So this chapter, I will talk about the double-strand DNA. You know, DNA structure is double-strand helix. This structure was reported by James Watson and Francis Crick in 1953. But DNA is not naked structure in animal and plant cells. Usually, DNA bind to histone and form chromatin. It results in packaged structure in nucleus in the cells. DNA sequences have genomic information, so a lot of researchers investigated all genome sequences in the cells. I show the DNA structure, especially this DNA is B form DNA reported by Watson and Crick. You know that DNA has four kinds of bases. One is guanine, cytosine, adenine, and thymine. And guanine binds to the cytosine and the adenine binds to thymine by hydrogen bonding. Its structure called as complementary DNA pairs. This hydrogen bonding is very important for them to bind specifically. So, in this slide, I want to consider about the length of DNA. DNA is packaged in nucleus in the cells, and this DNA length in one human cell is from 1.8 to 2 meters, very long and very big molecule. So please consider how long is the total DNA in one human. I want to give the hint to consider about it. One human consists of 33 trillion cells. And please compare the DNA length in one human, and the distance between the Earth and the Moon. It's about 380,000 kilometers. And in the case of around the globe, it's about 40,000 kilometers. Please compare each distance and the DNA length in one human. All of human genome sequence were identified Human Genome Project at 2003. As a result, various diseases can be understood at the DNA sequence level. Why is DNA sequence important for us? Because amino acid sequence of protein depend on DNA sequence. At first, DNA is transcribed to RNA, and then protein is produced based on RNA sequence. If DNA sequence is mutated, amino acid sequence is changed, and then mutated protein is produced. This mutated protein causes many diseases. The DNA sequence has the information of appearance and body and character and the future illness. It means that this human DNA genome sequence is very important for us. You know that the life information is stored as genome DNA. So DNA is responsible for passing the information to the next generation. Based on this information, we already enter the age of genomic medicine. In this slide, we want to consider about the difference of the DNA sequence. In the upper case, the differences in DNA sequence between human and chimpanzees is about 96% similar. Very similar. Just a 4% DNA sequence is difference. But you know that humans have a lot of character. So you know the highest and character and the quite difference between each persons. But difference in the DNA sequence between human is just is 99.9% similar. It means that the 0.1% DNA sequence is difference between the humans. Almost same, but a little bit different. Why we can see such a difference and DNA sequence in the human cells? So we show the example of 3 persons' genome information. For example, Mr. A and Miss B and Mr. C. This person has a mutation at a different gene position. It's called genome polymorphism. Genome polymorphism. Gene polymorphism means one base difference in genome DNA causes individual difference. This information is very important for the medical area. And this genome information promotes the genome medicine. Genomic medicine means medical care that enables prevention, treatment, and medication of individual disease based on genomic information. It means that all of the DNA sequence of human is very important for this medical area. And I want to show the example of this anti-cancer agent. You know that anti-cancer agent is very important for us to repair the cancer. But you know that if you get the if we get the anti-cancer agent, this drug has a heavy side effect. It's very heavy side effect. Very useful, but very toxic. But we can expect the side effect of anti-cancer agent, even if we didn't get the anti-cancer agent. We want to show the example. In the case of Mr. L, when A gets the anti-cancer agent A, there's heavy side effect. But if person A gets the anti-cancer agent B, and there's not any side effect, very effective to person A. It means that anti-cancer agent is very useful for Mr. L. And the genomic sequence is different to the B. In the case of person B, if person B gets the anti-cancer agent B, there's a heavy side effect, quite different effect between the person A and B. It might depend on the difference of gene type. In the case of person C, the genomic type of C is similar to A. It means that person C chose the anti-cancer drug agent B, even if C didn't try to get the anti-cancer agent A. It's very useful for us to get the anti-cancer agent. It means that genome sequence DNA is very important for us. But, and you know that the DNA sequence is very important for our character and the illness and the medical area and so on. But we don't know about the detailed information of the genome DNA. Even if you already get the all DNA sequence, why? Can you imagine that? Nobody knows about the exact answer, but we imagine that the DNA has not only double-strand helix, but a lot of local DNA conformation. For example, bending and the single strand, and there's a local conformation and so on. We imagine that such a DNA local conformation is very important for the DNA event. This is the DNA structure. Middle one is a typical B-form DNA conformation. It's reported by Watson Crick and James Watson and Francis Crick. And this DNA for B-form DNA is right-handed double-strand DNA. It's similar to the A-form DNA. But Z-form DNA is quite different to the A-form and B-form DNA. Can you notice what point is different of Z-form DNA comparing with the A-form and B-form? In the case of A-form and B-form DNA, this structure forms right-handed double-strand DNA. But in the case of Z-form DNA, this structure forms a left-handed double-strand DNA, quite different. Of course, all of DNA consists of adenine, thymine, cytosine, and guanine, but double-strand DNA is quite different. This is the information about the A form, B form, Z form DNA. B form DNA formed right-handed, and the Z form DNA formed left-handed. Why this double-strand DNA formed quite different structure? Because structure of glycoside bond conformation is quite different. In the case of right-handed double-strand DNA of B form DNA, sugar formed anch, but in the case of left-handed DNA of Z form DNA, sugar conformation is anch and thin. So I explain about the structure of what is an anch and what is a thin. This is the structure of the sugar and the bases. In the case of B form DNA, sugar, for example, adenine and cytosine forms such a sugar conformation. This conformation is called as anch adenosine and anch cytosine. But this conformation is not formed as a thin adenosine. So can you notice what point is different? Different point is the rotation of this bonding. Okay. If this base rotated to this side, this position of the adenine base is over the sugar. It means that there is a straight hindrance between the sugar and the base. It means that this structure is not unstable, and in the case of B form DNA, usually this anch adenosine is formed. But in the case of Z form DNA, purine formed anch formation. But in the case of pyrimidine forms the anch formation. But in the case of purine base, this purine base forms a thin conformation, even if there is a straight hindrance. This different structure causes right-handed and left-handed DNA. And moreover, we know what kind of condition is important for DNA to form the A form, a B form, and the Z form DNA. For example, such a CZ sequence can form the Z form DNA instead of the B form DNA. And especially in the case of high concentration of salt and base modification and some kind of protein induce Z form DNA from the B form DNA. And especially we know the Z form binding and Z form DNA binding protein. If this Z form binding protein binds to B form DNA, this protein can induce the Z form DNA. This is the X structure of such a Z form binding protein with DNA. This green conformation is Z form protein in ADR1. This is the name of protein, and this is the name of domain. And the Z alpha protein bind to the Z form CZ repeat DNA and induce a Z form DNA. It means that in our living cells, there is not only B form DNA, but Z form DNA in living cells. But the most important is that the function of such a local conformation of the Z-form DNA is unknown. Near future, we want to investigate the function of the Z-form DNA. I think that the most important is that in order to investigate the function of Z-form DNA, we should focus on each DNA binding protein. For example, in the case of B-form DNA, such a binding protein binds to the B-form DNA due to the specific DNA sequence, and as a result, this protein can control the transcription and replication and so on. And you know that we already reported that there is such a Z-form binding protein in the living cell. And this protein binds to the Z-form at the CZ repeat site. And of course, this protein has a lot of function in the living cells. It means that Z-form DNA has a function through such a Z-form binding protein. In the future, the function of Z-form DNA is reported, will be reported, I hope.